everybody, welcome back for another episode of Behind the Brain Neuroscience. I'm your host, my name is Julian Moreno, and I'm a current undergraduate student at the University of Southern California, and today I will be breaking down Neuroscience 101 for you, the physiological and functional components of the neuron, right? Because I know a lot of you are new to neuroscience, I felt like this video was necessary so you can start understanding the breadth and depth of what I talk about on this podcast and what other professors might be talking about. And I feel like in order to understand that, you need to understand neuroscience at the cellular and molecular level. And that's what I'm here to do for you today. I'm here to break that down for you and to give you a better understanding of that. So let's talk about the neuron and different components of it. Let's start off with, I think some basic vocabulary words, right? So everyone hears about the action potential. I believe action potential is one of the most talked about things in neuroscience. It's one of the most exciting things. You got the pun there, you got it. An action potential is an electrochemical signal that runs the length of an axon. So this signal is not only electrical, but it's also chemical, and we can see this here where I put down the definition for electrochemical signal, what an electrochemical signal is, and that's a signal that is both electrical and chemical. It consists of ions creating a potential and neurotransmitter that binds to a site-specific receptors. So it can either be excitatory or inhibitory. We're going to break that down for you right quick. So what I mean by a signal that is both electrical and chemical, this signal has voltage, right? Electricity running down the cellular membrane of the axon. But at the same time, there is also vesicles that are filled with neurotransmitter being sent down as well. And they're also made locally at the terminal buttons. However, it's both electrical and chemical. So whenever the electrical signal reaches the terminal, it excites the terminal, and then the terminal turns to release neurotransmitter, and it either excites or inhibits the next neuron at its postsynaptic binding site, right? So that's where it can either be inhibitory or excitatory depending on which neurotransmitter is inside of these vesicles. So, I talk about postsynaptic. Well, what does that mean, right? What's a synapse? So a synapse occurs when dendrites latch to the terminal buttons. However, there is no latching between. There's actually some space there. And there might even be a role player, which we will talk about later on. It's a different type of cell that's going to help this process occur. Sometimes it's there, sometimes it's not, right? So in order to give you guys a good understanding of what the neuron is, we're going to start off here at the terminal buttons. So I want you guys to think of the terminal buttons as the presynaptic end, meaning this is going to be the end of the connection that is sending the message, right? It's releasing the neurotransmitter. And this is exciting because, it's very analogous to like a mailman, right? He's going to deliver the message and he's going to go put it in your mailbox. And this mailbox can be a receptor on a dendrite, right? So I'm the mailman, which means I'm the terminal button. And I'm going to deliver this message, put it in your mailbox. And when you read this mailbox, when you read this message coming out of your mailbox, it's either going to be a good message, right? Whenever you get good mail, or it's going to be a bad message, signifying bad mail, which will be inhibitory. And neurotransmitters such as GABA, it's a major inhibitory neurotransmitter in the nervous system, will inhibit another cell compared to, let's say, glutamate, which will excite the other cell. So that's the terminal buttons for you, and I just want you guys to remember, it's like the mailman, right? The terminal buttons are delivering the message. They're delivering that electrochemical message to the next neuron, right? So coming from the terminal buttons, which are the presynaptic end, I'm going to teach you guys about the dendrites, which are here in pink, right? And you can see they have this long, elaborate branch-like structure, for good reason too, because you know, if you're receiving something, you want to be wide and you want to be able to gather more information. And the structure of dendrites actually differs depending on where the cell is located and the cell's function. Some neurons have more dendrites, such as Purkinje cells, and then others have less. So it's actually the structure of these dendrites is very elaborate, and it makes sense both functionally and for structural purposes as well. So let's break down what these dendrites are. So these dendrites, like I said, you want to see these guys as the receptors. It's the postsynaptic end, and it's analogous to a mailbox. So like I said earlier, your terminal buttons are going to be the mailman now, and the mail itself is going to be neurotransmitter, right? The mailbox is going to be your receptors, because it's actually receiving the message and taking it into the rest of the cell. So I really want you guys to get these analogies down, because these are really going to help you out when understanding what the dendrites do for you, and you know, their essential function and role in synaptic activity when the action potential is being transferred from one neuron to another. So it transmits electrochemical signal then to the soma. So let's talk a little bit about what's going on here. Basically what happens is these dendrites are going to take in that electrochemical signal. Like I said, if it's excitatory, it's then going to make its way to the soma. It's going to have to be strong, and it's going to have to come in at different angles in order for it to make its way to the soma. Once it makes its way all the way to the soma, which is the cell body here, right? It's this space in blue. This is where the control center is. It regulates cell activity and integrates signals coming in from the dendrites. I want you guys to think of the soma, the cell body, as the White House. It's calling all the shots. This is where everything's happening at. It's also, you know, very important for housing the nucleus, which stores genetic info and is the central area for neuronal protein synthesis throughout the rest of the cell. So if you need something down in the dendrites, up in the dendrites, because remember that's our postsynaptic end, you know, you're gonna need the nucleus to send new receptors up there. At the same time, you're also gonna need, you know, sometimes the terminal buttons are gonna need some proteins, and then also the axon itself and other parts of the neuron. So let's talk about this, right? So before we even get to the axon, we have the axon hillock, which serves as a bridge from the soma to the axon, so it acts as the neck, right? Imagine my head is the soma, you know, this big round bubble, and it has the brain in it. You can say that's a nucleus, right? And then here's the neck, and the rest of my body is going to be the soma. And you can, I could put my hands up here and say these are going to be the dendrites, and they're long and elaborate, and they're just ready to catch whatever signal's coming this way to me. So here at the axon hillock, like we said, it's the neck. It serves as like a bridge from the soma to the axon, and it's responsible for the all or nothing action potential, and it's where it starts. So basically, once this electrochemical signal reaches the axon hillock, it's like, hey, are we going to send down this message or not? And if we are, we're sending it full throttle, you know, there's no going back. And that's what I mean by all or nothing, meaning that, hey, axon hillock, we're going to send this message, boom, it's getting sent, and it's getting sent with no delays or nothing getting in its way. So let's talk about the axon itself. And well, what the axon is, as you can see here, is this green, this long green propagation that sends itself all the way down, connecting to the terminal buttons. I don't really want you guys to see this full picture here that I drew out. And I left some space in between, which is the cellular membrane, that little white space. And I really want you guys to understand that the axon carries electrochemical signal, right? This is where everything's being sent down through. And neurotransmitter flows down in vesicles. So it's flowing down from the soma, and it's going to make its way down to the terminal buttons. However, the terminal buttons do make neurotransmitter locally, because sometimes that process takes too long. So it's like, hey, we kind of need some neurotransmitter here locally whenever people are doing things fast, or there's no time for recovery. So that's the reason for local neurotransmitter synthesis, or the creation of neurotransmitter at the terminal buttons locally, meaning that it's made there, and it's made there for that reason, that sometimes you can run out. So let's talk about, like I said, the axon. Electricity runs down the cell membrane, so it runs down in this little white area right here, right up and down. And I want to say it's analogous to the freeway, because when you're on the freeway, you're hauling butt. You're going to be going, sometimes, I know, some of y'all, maybe you go the speed limit, okay. But still, in terms of speed, you usually go fastest on the freeway. And when you're on the freeway, it's all or nothing. When you're getting off of the freeway, now this is what we call the axon sending that message. Let's say your car, right? Like I drew out here, you're the car. And you inside, you're that neurotransmitter, and you're ready to exit. That means you're getting ready to go into the terminal buttons, and you're ready to make that exit and head to your final destination. So I really like this analogy of, the axon's the freeway, because once you get off the freeway, well, then you're ready to make, you're exiting, and you're ready to make your turn into your final destination. Which is actually pretty interesting, because like I said, you yourself will be the neurotransmitter, and yeah, you're either gonna go to someone's house and excite them, or you're gonna show up at somebody's house and you're gonna make them angry, or you're gonna inhibit them, from them going out. So now that we've talked about the axon, I want to talk about this orange layer that's surrounding the axon, and this is called the myelin sheath. And what the myelin sheath is responsible for, well, first off, it's a fatty lipid substance, right? And it wraps around the axon, and it's wrapped by these cells called glial cells, which, remember, I told you earlier was going to be one of those cells that's going to be maybe a key role player in helping that synapse occur as well. But it also plays a role and function as like a construction worker, and it wraps around this fatty lipid substance. It wraps it around the axon continuously, and what it serves as is it serves as an insulator. So my analogy for you here to remember the myelin sheath is that it's the rubber surrounding a wire, and the wire for you here is going to be, your axon, and that's where that electrical signal is going to be sent down, right? That action potential is going to be sent down, and in order for you to keep that action potential going without losing its strength, because as we know, energy will disperse over time and distance, and it won't be as strong anymore once it's going through once passed through a certain distance. You don't want to move your leg, and then you're not able to move your leg because the action potential that ran from your brain all the way down to your not from your brain, actually it occurs from your spine initially. But if you want to make some sort of movement, obviously, of course, it's first going to start in your brain. It's going to go from your brain to your spine and then to your foot. Well, you don't want to malfunction and only move your foot a certain amount. You want to move your foot with your full strength, and that's why it's important for the myelin sheath to be there, because it keeps the action potential strong, and it allows it to travel distance and over time, right? And there's these little dips that I drew here in the myelin sheath, and we call these the nodes of Ranvier. And what happens at the nodes of Ranvier is it's a point of entry for ions along the cellular membrane, right? And what I mean by the point of entry, basically meaning this is where sodium's going to enter, this is where potassium's going to leave, and it's going to be a depolarization of the cell. So it's the net flux of sodium and potassium to continuously depolarize the cellular membrane, right? So here we're going to have sodium, let's say on this side is the inside of the cell, right? And this side's going to be the outside of the cell. Sodium is going to make its way into the cell, while potassium is going to make its way outside of the cell. And when this is happening, you're depolarizing because you're taking depolarization, which means two opposite charges, right? You have a net, positive voltage and you have negative voltage. And the negative is going to go to the positive is going to go to the negative. In this case, potassium to the outside, sodium to the inside of the cell. And when this is happening, that's when you have the depolarization of the cell, and that's going to create this little propagation wave that we call the action potential on a graph, right? And this is going to happen over time as well. And in order to keep the strength of the signal going, that's why it happens at different spaces along the axon, because it's like, hey, we need a little reboot system, and the nodes of Ranvier are going to serve as your charger or your reboot system, right? To keep the signal strong over time and distance as well. So the myelin sheath and the nodes of Ranvier, if you don't remember anything about these two, the one thing I do want you to remember is that they help keep the signal strong over time and distance without letting the energy disperse or be lost in that time or space continuum when all this is happening. So that is going to give you the summary of everything that happens. So I'm going to break this down one more time for you guys, right? We're going to start off here at the terminal buttons, right? Because I feel like this is the best way to like explain what's happening. Terminal buttons are going to release neurotransmitter. They're going to act as your mailman. They're going to put the mail in the box, right? This box is going to be your dendrites. And the dendrites are going to receive this chemical message, this electrochemical message. Don't forget about the electrical part. And that message is going to be sent down to the soma, where it's going to decide, hey, is this an excitatory or inhibitory message? Are we going to excite the next neuron? Are we not? And then once it decides, hey, we're going to excite the next neuron, what's going to happen is it's going to send the axon hillock is going to make that decision, and it's going to serve as the bridge from the soma to the axon for that all or nothing action potential to occur. The action potential is going to run the wavelength of the axon. It's going to run all the way down the axon, right? All the way down it. And through its journey, it's going to have to stay strong over time and distance. So the myelin sheath and the nodes of Ranvier are going to play as key role players here in keeping that signal strong. So that way, you know, when you want to move, you can move. A lot of neurological diseases happen because this myelin sheath is not, the proteins responsible for creating the myelin sheath there's some sort of malfunction there. So then that way the neuron's not covered in the myelin. So then what happens is the neuron's not able to fire and like keep that, well, it's able to fire, but then that action potential, that signal is going to lose strength over time and distance. And that's why when it reaches the next cell, you know, it's not strong enough to excite it. And that's when you have people that have diseases such as, not Alzheimer's, but like Parkinson's disease, etc., you know, where they have a shuffling gait, or diseases where people aren't able to move. You know, there's just a lot that goes into that, and it's really exciting to see and understand. So to get back on track, you know, the signal's gonna go down the axon, and what's gonna happen once it makes its way down the axon, it's going to end up reaching the terminal buttons, and once it reaches the terminal buttons, that's when it's going to disperse again, and that's where, the electrochemical signal will reach the end of its journey, and, it's going to excite or inhibit the next neuron. So that is the basic, breakdown of the neuron for you. I hope you guys enjoyed this video, and I hope it really helps you guys out with your understanding of the neuron. If you have any questions, leave your questions underneath, the comment section. Yeah, I just hope you guys learned something from this, and stay curious. There's always so much to learn, and that's one thing I've had to realize as an undergrad. Obviously, it's like I'm dumb right now. I just have so much to learn still, even though I've got this down. Well, then I've got the cellular molecular level down. Well, guess what? There's another level after that, another level after that. So just keep gathering this information, keep your understanding, and understand how to, create relationships between this cellular and molecular level to a systems level, and know what's happening at both spaces simultaneously, right? Because it's like a microscope, sometimes you're going to see things small, sometimes you're going to see things big, and the small things are going to be what's going to affect the larger things in the grand sense of things, right? In the grand sense of the whole entire scheme. Well, thank you guys for watching this episode of Behind the Brain Neuroscience. You guys keep on. Thank you so much. I can't thank you guys enough for watching these videos. It means a lot. Keep learning, keep being interested, and yeah, I got a lot for you guys coming soon. Thank you for being patient. I know I haven't dropped in a minute, and this was BTB Neuroscience, and we're out.